All right, so today we're going to talk about an area of study design that's really closely aligned to what we've been talking about, and it doesn't add too much to the whole story, but it does sort of highlight how this inheritance stuff, yeah, it's complicated as it is, but it's more complex. It's not as straightforward, and that makes sense because we have all of these different people in the world, and the genes don't just inherit as simply as we're talking about, right? And one of the reasons why is what we call linked genes. Okay, so in the study design, we're still looking at that same area of study design: pedigree charts, genetic cross outcomes, genetic decision making. But we want to talk about the inheritance of two characteristics as linked characteristics and the biological consequences of crossing over for linked genes. So we've actually till now we've been talking about independently assorted characteristics. All right, and what that just means is that, let's say we're having a dihybrid cross, so two different um, genes, um, but one gene would have been on, let's say here could have been the gene for eye color, all right, and that could have been on chromosome number 2. But then if we were to inherent or talk about the inheritance of hair color, that could be on chromosome number 5. We've got two different chromosomes, so they are independent. So these two genes are independent of each other. All right. Linked genes, on the other hand, let's say we have a certain trait. It might be on chromosome number 4. It might be the trait for height. All right. But then we could also have on chromosome number 4 the trait for hand width. Okay, in this case, both of those genes are on the same chromosome, and so we call that linked genes. Okay, and then from this, we're going to go and touch back on to crossing over. So I want you to try to explain this. So we have two heterozygous, so two different alleles on their chromosomes, two heterozygous horses that are crossed. Both have the dominant traits of black mane and brown coat, and let's pretend that they have 16 fold, just so the numbers are all really nice, okay? Um, the breeder was very knowledgeable about dihybrid crosses, and he believed that the offspring would be 9 out of 16 black maned, like this, and brown coated, 3 out of 16 black maned but tan coated, 3 out of 16 chestnut maned but brown coated, and 1 out of 16 chestnut maned and tan coated. Okay, so where black maned is dominant over the chestnut maned, and brown coated is dominant over the tan coated. Okay, um, why do you think that? Because he thought that because we're talking about the heterozygous dihybrid cross, which, as you know, is a 9 to 3 to 3 to 1 ratio. That's that golden ratio of the heterozygous cross. But the ratios actually looked like this. We had 11 out of 16 that were this fully dominant horse, all right? We had 3 out of 16 that were the fully recessive, okay? But then we have a very tiny proportion of the ones that were black maned black maned and tan coated, and chestnut maned and brown coated. These two guys, all right, we have a very tiny proportion, this 1 out of 16 like this. We want to look at why. Why did this happen? Before we go on to that, though, have a look at these ratios. Let's add these two together. What do we get? We get 12 out of the 16. And then if we added these two together, what would we get? We get 4. So a ratio of 12 to 4 is actually the same as 3 to 1 ratio. And what do we know? Where do we usually see the 3 to 1 ratio? So this is an example of a dihybrid cross with linked genes. And what do linked genes mean is that, just like I showed you before, we're going to have two genes that are found on the same chromosome. So, for example, the main color might be up here, and then the coat color might be here. So they found together and they're going to be inherited together. So that's why we actually write the genotypes, for example, of the heterozygous parents, we'd write them as big M, big C together like that. We're showing that these two are inherited together, right? And then over the top of little m, little c. I'm just going to put a line there just so we know that they're the recessive versions, all right? And we're crossing that with two heterozygous, so big M, big C over little m, little c. Okay? We're going to use Punnett squares to show the expected percentages of the phenotypes of the offspring. Okay, so in this case, when we're doing the Punnett square, if we were to look at one of these parents, what gametes can they give? So these two genes, remember, they're nice and close to each other, and they're going to be inherited together on the same chromosome. So that means that this parent can either give big M, big C, or little m, little c. So there we would write big M, big C, little m, little c. And this parent would be much the same. So big M, big C, little m, little c. Okay? And so when we put the offspring together to find out the possible offspring, we just find big M, big C there over big M, big C. And then here we'd have big M, big C over little m, little c. And then we'd have big M, big C over little m, little c. And finally, we would have little m, little c over little m, little c. All right? And how good is this? It essentially ends up being just the monohybrid cross, that nice and easy cross. So when we do the data analysis, the possible genotypes we have are the double dominant, homozygous dominant. We would have the heterozygous genotype, and we would finally lastly have the homozygous recessive genotype. Okay, for the phenotypes, hopefully you can identify that, hey, we've got at least one version of the dominant in here, so we're going to have the dominant phenotype, which was brown coat and black mane. All right, and this homozygous recessive is the only one that's going to be able to give us the tan coat. What was the last main color? And of course, chestnut. Chestnut mane. All right, if we look here, we've got a ratio of 3 of the dominant phenotype, the fully dominant phenotype, 3, to a ratio of 1. Cool. Look at that. 3 to 1 ratio. We go back here. Oops. We go back. And we've got pretty much the same 3 to 1 ratio. But that's not taking into consideration we've got a tiny quantity of these weird intermediates. Let's talk about why, all right? And the reason why is because of crossing over, okay? So going back all the way back to meiosis, crossing over is that process where we get the swapping of sections of the homologous chromosomes during the prophase 1 of meiosis, all right? So what would happen is if we had these as let's say that we're talking about chromosome number 2, or it doesn't matter which one, but we have the maternal chromosome and the paternal one too, so the mum's and the dad's one, and if we had a crossing over event happening, what we would get is something like this looking at, okay? So bits of the chromosome from the mum swapped with the chromosome from the dad, okay? So for example, let's say that these two genes, the main color was up here and the coat color was down here. If that happened and we had the big M, big C here, and then, but here we had the little m, little c, all right? Those big M's and big C's, they wouldn't be no longer found on the same chromosome. So here we would find the little m. Down here we would find the big C. Here we would find the big M, but here we would find the little c. All right, and that's how we're going to get those horses with the black mane, tan coat, and chestnut mane, brown coat. Okay, it is important to realize that the only reason this happened is because these two genes were far enough away on that chromosome. If, for example, that coat color gene was actually found down here, like we originally had put them, then crossing over, even though it happened, the genes would actually carry over together. Okay, so just summarizing all of that, it is important to understand that meiosis outcome due to crossing over. So if we don't have crossing over occur, everything looks really nice, but unfortunately, it's not what actually usually happens. But we pretty much have two genetically identical cells on one side and two genetically identical cells on the other side. But what usually actually happens is crossing over, where we get, because of that crossing over event, we get a swapping of bits of chromosomes and often a splitting of genes that are linked. Because remember that on a chromosome, you might have a few hundred, few thousand genes, depending on the size of the chromosome. And so, um, a lot of those genes are going to swap over due to that crossing over event. Okay, so genes on the same chromosomes will be inherited together unless separated during that crossing over. And like we just looked at, the further apart the genes, the more likely they will get separated by the crossing over event.